All right, in section 4, we'll be looking at what's called the adaptive immunity. This is different than your innate type of immunity. Adaptive is also known as your specific immunity, and it involves ability to recognize a particular substance or pathogen or toxin that enters the body, respond to it, and then remember it, so that if in the event you come across the exact same pathogen again, your body can adapt very quickly to it and initiate an immune reaction very quickly and very strongly. Now, the two main types of cells that we'll be talking about for the adaptive immunity are the T cells or T lymphocytes, and that involves a process of cell-mediated immunity, and then your B cells, and that involves a process of antibodies or antibody-mediated immunity. Now, one of the words that we'll be mentioning a lot through this section is the word antigen. That comes from the two words antibody generator or antibody generating. And there's two main types of antigens really referred to in the body. The first one is called a self-antigen. This is actually a marker or a molecule that's produced by the body's cells, and it's really used to help identify the cell as belonging to the body itself. It creates a signal so that your defense mechanisms, your immune response, will recognize your own cells as being part of the body, so that you don't have a autoimmune type disease, so that your body doesn't start attacking itself. Your body's white blood cells have to have the ability to recognize and to differentiate between something that's a self-antigen. Say, for example, your liver cells would have a self-antigen on there so that your body knows not to go and attack the hepatocytes, the liver cells, versus a non-self antigen, and that would be anything that doesn't belong in the body, such as maybe some kind of microorganism or virus or some type of parasite. Another word that goes right along with antigen is what's called the antigenic determinant. Sometimes you just see that referred to as an epitope. This is the actual site on a given antigen that would be recognized by your white blood cells or your lymphocytes in this case. And so the picture kind of shows you some kind of antigen. Maybe this is a type of bacteria, some type of cell, bacterial cell or parasitic cell, and it has many different markers on the cell. And all of these, the whole thing, if you talked about the whole thing, could be considered to be an antigen, but the individual markers on that surface would be the epitopes. This is actually what's causing the reaction to stimulate your immune system. So we mentioned a little bit about self. So the non-self type antigens, that's probably what we think of most of the time when we hear the word antigen. And these are really just foreign antigens or non-self, things that are not produced by the body, such as the bacteria and viruses and other types of microorganisms, parasites or fungus. But it also could be things like pollen. If you ever have pollen allergies or pollen is an antigen, would be considered an antigen because it tries to initiate an immune response in the body. Some people are allergic to cats or other animals. Mites produce antigens in the waste and the feces. Certain foods have antigenic type components to certain people, and then some people are allergic to certain types of drugs or medications. There's a growing number of people that seems to be allergic to penicillin, and so that creates an overreaction of their immune system. So that would be an antigen, a foreign type of antigen. And we've already mentioned the cell types we're going to look at. Cell-mediated is all about what's taking place with the T cells. Antibody-mediated is what's going on with B cells. So given that, let's review a little bit about B and T cells. Remember that they originate in the red bone marrow. That's where the stem cell is located, is inside the red bone marrow, and that's where you produce all of your blood cells, including these two lymphocytes. The difference here is that they have to be inside of their what's called the primary lymphatic organ to finish the maturation process, to finish the developmental process. Now, for B cells, that's easy because they complete their development in the bone marrow. So they originate in the bone marrow, and their primary lymphatic organ is still the bone marrow. But for T cells or T lymphocytes, their primary lymphatic organ is actually the thymus gland. And so once they leave the bone marrow, they enter in the bloodstream and they make their way, they'll circulate in the bloodstream. And when they come to the thymus gland, that's where they're going to find that hormone thymosin, and that helps in the developmental process to finish the development and maturation of the T cells. Part of what's going on in the thymus gland is something called tolerance. The book definition is listed there for you: a state of unresponsiveness of lymphocytes to a specific antigen. Inside the thymus gland, your thymus is trying to screen T cells, trying to check to see, which ones are working correctly. If they seem to be self-reactive, if T cells seem to be, if they respond to a self-antigen, then you need to get rid of those, because that means that they could go around and respond to your own, body's antigens. And you don't want that to happen, because that could lead to an autoimmune disease. If you have T cells that are non-reactive, they just don't react to anything, then they get screened out. And so part of what tolerance is in the thymus gland is to try to test for lymphocytes to see whether or not they respond to certain situations. Now, let's begin looking at what has to take place for lymphocytes, for your T cells to actually work. Step one is they have to be activated. And so you can just kind of think of this as like a switch. They're either turned on or they're turned off. And so most of the time your lymphocytes are just going to be in a dormant type state. But whenever they're presented with an antigen, some type of foreign substance, we're kind of talking about that non-self or foreign antigen. When they're presented with that antigen, then they will become activated. And in the picture, you see a special type of lymphocyte, a dendritic cell. It's called a dendritic cell because of these long extensions that it has. It kind of looks like dendrites of a nerve, of a neuron, but a dendritic cell is a white blood cell, a lymphocyte, that will present an antigen to other lymphocytes, to your T cells. And so in this particular case, this dendrocyte has picked up and is binding to some kind of antigen in a receptor, and you see it presenting it to two different T cells here. Well, that process involves receptors. The process of presenting an antigen to a T cell actually involves different receptors that are on the membrane of the cells. And you have a couple of different categories of receptors. Now, the receptor main category here that we're looking at is something called the major histocompatibility complex, or MHC type receptor. And we're going to look at two different classes. Class 1 is the most common. It's found on pretty much all of your nucleated cells that have a nucleus. So, there's not that many cells that don't have a nucleus. Your outer epidermal skin cells, those are dead cells, so it's not really there. Your red blood cells, once they're mature, they don't have a nucleus. So most of your cells are going to have a nucleus, and that's where you're going to find the class 1 type receptor. And the process, you can kind of see in the diagram here, you've got some kind of cell here that has taken some type of antigen on the inside of the cell. So for example, think about a virus. The virus that goes in, attacks a cell from the inside. This cell here that you see has takes in some of that antigen. It processes it, it sends it through the rough endoplasmic reticulum. It puts it on a little vesicle to package it into a vesicle, and then it displays it on the membrane. What this is trying to do is to alert other cells that there is, a virus infected cell in the area, and that the diseased cell needs to be destroyed. And that's one of the key points here, is that the diseased cell, whatever cell is presenting this, must have some kind of virus particle inside. And it's trying to alert, the other white blood cells in the area that this particular cell has a virus inside of it and it needs to be destroyed. So the outcome here is to destroy the diseased cell. The second type of major histocompatibility complex receptor is just called class II receptors. And these are found on the surface of antigen-presenting cells. So your immune cells, such as your B lymphocytes, your B cells, would have class II receptors. Some of your other white blood cells, such as your macrophages and monocytes, these are your phagocytic type cells. They would have the class II receptors. And then also your dendritic cells. Now, this particular scenario kind of starts the same way as a class I. You've got some kind of pathogen, and either the pathogen or a part of the pathogen gets taken in by the cell. It puts it inside of a lysosome. It starts to break it up, and then it packages it and puts a portion of that antigen on the surface as a signal. It displays a portion of the antigen on the surface. But the key difference here is that this type of presentation is going to stimulate other immune cells to initiate an immune response to try to get rid of the antigen, try to get rid of that pathogen without destroying the presenting cell. So if you have a class II receptor, you're able to present and activate your immune system without destroying this cell. And that's different than class I, because remember, class I is going to actually destroy that presenting cell. Now, once the immune system cell is activated, you go through a process called clonal selection. You basically select the cells that you want to make more copies of. That's clonal selection. T cell clones that specifically bind to the antigen are activated. In other words, you have many different types of T cells, but only the ones that you really need are actually activated. Many other T cells you don't need to activate because maybe they are specific to a different pathogen. Maybe you have, a particular virus infection, maybe the flu virus. Well, the T cells that are specific to a cold virus or some other virus, they're not needed at this point. You need to activate the ones that will respond to the flu virus. And so those are the cells that have to be cloned. So that's clonal selection. You select for the clones, or you select for the T cells that need, that you need to make more copies of. Then you go through the process of proliferation. That means making more copies. Now that you've selected the ones you need, you proliferate them, you make more copies, and then they differentiate into effector T cells and memory T cells. It's the effector T cells that will go out and actually, fight off the infection. They're the kind of the frontline cells. They're, destroying the invading pathogen or the infection. The memory T cells retain the memory of the infection. They retain their memory of that antigen, so that in the event you're presented with that antigen again, they have a memory for it, and they can ramp up the immune response very, very quickly. We'll see some information on that, how that works a little later. Now let's look at a few examples of some of these T lymphocytes we've been talking about. You have helper cells. These fall into a category called regulatory cells. They help to regulate activities of the immune system. So you have those that are helping the process. These are helping because they help to activate other immune cells in the area. So, once you've gone through the activation and clonal selection and differentiation, you've got helper cells that help to kind of help the immune response by activating other cells, other white blood cells in the area, including macrophages, other T cells, and other B cells. But it's the cytotoxic T cells. The word up here, toxic, is telling you that these cells are toxic to other cells. These are the cells that will directly lead to the breakdown or the destruction of the infected host cell. So they're the ones that will kind of, as you see in the picture here, you've got some kind of activated T cell here. This is the target cell. It's got a class I receptor, which means, the target cell here is going to be destroyed eventually. Probably it's got some kind of virus infected. It has a virus that has infected this cell. It's put that on a class I receptor. You've gone through the steps of activation and clonal selection, and now here you've got this activated T cell that recognizes this antigen, and it's cytotoxic, so it's going to actually release enzymes, chemicals inside of this cell that will break down this cell. Right? It broke down this cell, this is one of your cells, but remember, this cell had some kind of virus that you had to get rid of. And so there was no virus cure here, so it has to get rid of that cell. If you get rid of the host cell, you can get rid of the virus. Section 5 looks at antibody-mediated immunity, so we need to talk a little bit about antibodies. This really is a very effective means to getting rid of extracellular antigen, antigens on the outside of the cell, including bacteria, even some viruses that haven't penetrated a cell yet, protozoans, fungi, parasites, and even toxins whenever they're outside of a cell. So we'll be talking about antibodies. Make sure you know that the term immunoglobulin also refers to antibodies. The process leading up to antibody production is very similar in B cells as it was in T cells. So remember, antibody-mediated immunity is all about B cells, whereas the cell-mediated immunity was about the T cells. Well, the beginning steps are pretty much the same, right? The B cells have to become activated. How do they get activated? Well, they have to respond to an antigen. They had to be presented with an antigen. And so in the little picture here in this diagram, you see that you have a B cell. It's got a receptor on the membrane. There's some kind of antigen here, and it's going to, and it's got a class 2 type receptor here. This is class 2, which means, it's not going to destroy this cell. It's going to help activate other cells in the area. And so once the antigen binds to the receptor, this class 2 type receptor here is going to be ready. And so now it's what is referred to as sensitized. It's ready. It's almost activated now. And then once it binds to another helper cell here, a T cell, it can activate this helper cell. That in turn causes your B cells to start its process of activation. And so now you have the B cells that start to divide. The B cells start dividing. They're going through the proliferation and then the differentiation process, just like the T cells did. And so the first thing, they're going to start going through division, prophase, metaphase, anaphase, telophase, and all that. And they will differentiate into memory cells, just like you had memory T cells, you're going to have memory B cells. And then the cells here, instead of having T cells here, or just regular B cells, these are referred to as plasma cells. Now, it's the plasma cells that will produce antibodies, produce and release antibodies. So let's look at antibodies for a few minutes. Antibodies, also known as immunoglobulins, they have a variable region and they have a constant region. Remember that antibodies are proteins, and so you're dealing with a sequence of amino acids. And so the variable region is always found on the tip or the edge, up here at the tip. This is where the antigen needs to bind to. This is called a variable region because these are highly variable between one antibody and the next. Think about how many different antigens you could have. I mean, you've got millions and millions of different possibilities of antigens, and so these variable regions need to be highly variable so that they can bind to many different types of antigens that you're exposed to. The constant region is pretty much the same, so it doesn't matter really which antibody you look at. The amino acid sequence in the lower portion of the Y and all the way down is pretty much conserved. So the amino acid sequence doesn't really change very much in the conserved region or the constant region. Now there's 5 major classes or groups of immunoglobulins. Let's look at each one of these. IgG, immunoglobulin G, this is referred to as the main antibody. It's the most common. It's about 80 to 85%. If you did an antibody screen or titer and found out, how many different types of antibodies or how much of each antibody you had, you would find that you would have most of your antibodies would fall into this category. This is found in your serum, your plasma of the blood, and it has 2 distinguishing features. Is that it has the ability to cross the placenta. And so this is a relatively small antibody that has ability to cross the placenta that gives the developing baby, the unborn baby, some protection, some immune protection while it is developing inside the mother. So that's very important for the baby's immune system. It also has the ability to activate the complement. Remember, we talked about the series of proteins in the innate immune system. And so this, once it's activated, once a B cell is activated and it goes through its process, it produces the plasma cells. The plasma cells will produce IgG, and IgG can actually activate that complement cascade to help in your innate immune system. IgA, number 2, this is found only in about 15%. That's its concentration. About 15% of the antibodies would fall into this category, but you find this where you find mucous membranes. So mucous membranes associated with the eyes, around the, it's found in the tears. Mucous membranes associated with the mouth, it's found in the saliva. Any other locations you find mucous membranes, usually towards the openings, around the openings of the body, the GI tract, the reproductive tract, the urinary tract, any of these areas that have mucosal defenses, you would find IgA. It is again also found to help to give newborn babies some added protection, because it's found in the colostrum. That's the breast milk, the first type of breast milk that the mother produces. And so that colostrum has a relatively high concentration of IgA. And so that is transferring some what's called passive immunity. We'll talk about that in a few minutes, from the mother to the newborn baby. And that helps to kind of boost their immune system. IgM, this is also found in the blood or in the plasma. It is a free-floating type of antibody, just like the other two were. Only about 5 to 10%. This is common in agglutination type reaction. So it's common to find this where the mechanism that this works is to clump together different toxins or pathogens or virus particles. You clump all of those together, and then they don't function. So that's kind of how it works, how its action is to work. The other distinguishing characteristic about IgM is that it is the first produced during an infection. So you get higher concentrations of this whenever you have some kind of infection. IgD, immunoglobulin D, this is not directly found in the serum, only because it's not free floating, right? It's not freely passing through the blood serum or the plasma. Instead, it's found directly attached to the surface of the B cells. So it's on the cell membrane of your B lymphocytes. And that way it can act as a receptor. So it binds to antigens to activate B cells and then to activate other immune cells. Only less than 1%, so it's not as common as the other antibodies. The last one, IgE, immunoglobulin E, very low concentration of this. This particular antibody is, when it's released, will bind to mast cells and also basophil cells. These help to produce your inflammatory response. And so these are helping to release chemicals like histamine and other chemicals that cause allergic type reactions. And so, so if you have allergies, sometimes you have an overproduction of IgE. Now, IgE causes your mast cells and your basophils to release the histamines, and that's why you would have to take an antihistamine to try to slow down some of these inflammatory responses. You also find IgE or higher concentrations of IgE whenever you may be infected with some type of worm parasite, such as a roundworm or tapeworm or some type of pinworm, and you have this particular antibody has the ability to bind to and attack and recognize the types of parasitic worm pathogens. Now, the next section we'll talk about how do these antibodies actually work? What are the processes where these antibodies actually, actually fix a problem? So the first type is agglutination. This is the first example of an action of antibody. It's called agglutination. It just means that it binds to, kind of causes a clumping reaction. If you, like in the picture here, you've got some type of antigen maybe here. Maybe it's a virus particle, maybe it's a toxin, whatever. Here you've got some kind of cell, maybe it's a bacterial cell. You stick a bunch of these antibodies together and you clump all these different target cells together, and that just kind of it helps in the process of phagocytosis. You're trying to help your white blood cells. So your antibodies are there to assist your cytotoxic cells and your macrophages and your other cells that are going to come along and hopefully get rid of some of these bacteria or virus particles. So this clumps everything up and makes it a little easier for your white blood cells to come along and go through the phagocytosis process. Opsonization, this is similar, has a similar effect to the agglutination. It's not a clumping reaction, but basically the antibody, in this case they're showing IgG, antibody binds to a particular target cell, a particular pathogen, and it coats it, and then that makes it easier for phagocytosis, right? These two join together so that you're binding the antibody with the pathogen, but also the antibody binds with the phagocytic cell. And so you kind of create an ideal situation to try to help that phagocytic cell engulf that particular pathogen, whether it's a bacteria or some type of microbe. Neutralization is what you generally refer to when you're talking about getting rid of some kind of toxin. There's also complement activation. Remember, we talked about IgG, and so one of its actions is to actually start the process or activate those first proteins that are involved in that complement cascade system that will eventually lead to the MAC, or the membrane attack complex. And so remember, those, part of the innate system, those complement proteins are usually inactivated until they're kind of turned on. And so here you have a scenario where the IgG antibody actually stimulates and activates the complement to begin its process. Then the last thing we've already mentioned here, like antibody IgE, helps in the process of inflammation. And it does that by binding to some of those cells that are storing up those inflammatory chemicals, such as your basophils and your mast cells. Once the IgE binds to these cells, it starts to release chemicals that help in the process of inflammation. Now, the last couple things we'll look at, there's a graph here that I wanted to show you that illustrates and talks about the difference between the primary and the secondary response to an antigen. The primary response is the first time that you're exposed to a particular antigen. And your secondary response is the second time. And so there's some differences. There's three main differences that we'll look at. So for example, what is showing up here is what's happening with the cells. And so you have a B cell here that would be, it's turned on, it's activated whenever you're first exposed to a particular antigen. It's eventually going to start dividing. Remember, it differentiates to memory cells, and that differentiates to plasma cells. It's the plasma cells that make the antibodies. Well, if you graph that, down here at the bottom, you see the little graph here, and you've got time along the bottom, the x-axis, and you got the magnitude or how strong the response is on the y-axis. And then you mark the starting time here, the first exposure to the antigen. That's the first time you come in contact with it. You can see that it takes a certain amount of time till you get your response, and then it goes down. Compare that with the second time you're exposed. Maybe you come into that same exact strain or that same exact pathogen a second time, but notice there's three differences between the first time you're exposed and the second time you're exposed. One thing that's different is that you have a shorter response time. The first time you're exposed to an antigen, it may take up to 2 weeks to build up enough antibodies that you can actually have a response. But here, the second time you're exposed to that same antigen, it may only be a few hours or maybe a day or two to actually have a response. All right. Notice also that the response height is greater, the magnitude is greater the second time. The first time you build up, maybe a week or two, and you start building up some antibodies, but notice that you have a much greater response the second time, much higher response. So the magnitude of the response is greater the second time you're exposed to the antigen. And then lastly, notice how long your response lasts. The primary response, it doesn't really calibrate it. You don't have little numbers down here, but you can kind of see that the primary response must be relatively short. But in the second time you're exposed to it, it's higher, but it also lasts much longer. And again, we don't really have a definite time factor on the scale here, but you can see that that secondary response is much longer than the primary response. Now, the last topic I'd like to look at is the memory that is developed for your immune system. And there's really two main categories. If you'll take a look at figure 20-23, there's two main categories: active immunity and passive immunity. Active immunity you can think of as when your body is actually producing antibodies on its own. And that can either be through a naturally acquired activity. In other words, you're exposed to a particular antigen, and that causes your immune system to activate and start making antibodies. Or you can be artificially activate your immune system, and that's through the process of vaccination. Naturally exposed, in other words, you pick up a virus, a cold virus, or maybe even the flu virus, and you're exposed to that infection, and then you build up immunity toward that particular strain, and that gives you longer lasting protection. Artificially active immunity, that's just when you're vaccinated for it. Hopefully you get the flu vaccine, and then you have some protection, or at least partial protection, from the actual flu virus, and that's because you build up memory cells, and that gives you a better immune response the second time you're exposed to that antigen. This is different than passive immunity. That's whenever you transfer just the antibodies from person to person. And so you don't build up the memory. There are no memory cells that are formed, and you don't have that same type. You won't have that same type of secondary response like we looked at on the previous graph. There's really two ways that this can be achieved. Passive immunity can still be naturally acquired. And that's when we talked about, some of the antibodies like IgG can cross the placenta, and then antibodies that can go through the colostrum. And so that's going from mother to the developing baby. That's a possibility of transferring the antibodies naturally through a passive way. It doesn't give a long-lasting protection. The baby has some protection, but it's not very long-lasting. And but that's because there's no memory cells. There's no actual activation of the recipient's immune system. You can actually also receive injections of antibodies, and so you can artificially transfer an antibody from person to person. You see that sometimes with antivenoms. And so, if you're bitten by, if you're stung, you have some type of anaphylactic shock, or if you're bitten by a snake that has poisonous venom, then they use an antivenom, which is basically antibodies that would react to that venom and help to neutralize the venom in the person. And so that's an example of a passive immunity where you transfer antibodies to another person. And again, you don't have any type of memory cells that are formed, so you don't have any long-lasting protection.